Hello friends, welcome back to my channel, and today in this video we are going to study about how we can use transduction to map the bacterial genes on a chromosome. So just a revision of the method transduction. We already learned about this method, that when the gene transfer takes place via bacteriophage, we call it as a transduction. So what happens in the process? This is bacterial cell 1. When any bacteriophage attack any bacterial cell, it will insert its DNA material inside the bacterial cell. Then it will replicate inside the bacterial cell using the bacterial cell machinery, and during which it will degrade the bacterial DNA as well. Now it will pack its own DNA before release from this bacterial cell. But when it will pack its own DNA, very rarely, but it happens that it can pack by mistake the bacterial DNA. So when such bacteriophage with the bacterial DNA will get released from one cell and will infect another cell, what it will do? It will insert the bacterial DNA which it packed into the second bacteria. This is second bacteria, and this black DNA is the DNA from bacteria 1. So from the first bacterium, it transferred the DNA by mistake to the second bacteria. Now this DNA will get integrated inside the bacterial chromosome. So that's how when the gene gets transferred from one bacteria to another, we call this method as a transduction. So when any bacteriophage attack bacteria, two pathway it can follow. One is lytic cycle, another one is lysogenic cycle. What is lytic? Lytic means cell degrade. Lysis, the breakdown of the cell. That means killing of the bacterial cell. So what it will do? It will infect any bacteria. It will insert its DNA inside the bacterial cell. Then it will replicate. During replication, it will break the bacterial DNA also, and then it will pack its own DNA. But by mistake, sometimes it can pack the bacterial DNA as well, and it will break the bacterial cell and will release in the environment and will infect another bacteria. So this is a lytic cycle. Sometimes it can follow the lysogenic pathway. What will happen in the lysogenic pathway? In the lysogenic pathway, it actually don't kill or degrade bacterial cell. What it do? It insert its own DNA inside the bacteria, and this DNA will get incorporated into the bacterial chromosome, and it will remain in this phase only. This phase is called prophase, the phase where the viral DNA remained inserted into the bacterial DNA, and it will replicate as the bacterial DNA will replicate. The process of replication of viral DNA will continue in the same way as a normal process of bacterial chromosome DNA replication. When the environment becomes favorable, it will shift from lysogenic to lytic cycle, breaking the bacterial cell, and will get released into the environment. So the bacteriophage have two alternative life cycles, lytic and lysogenic. Lytic means when it kills the bacterial cell. Lysogenic means when it remains inside the bacterial cell. Now transducing phase means the phase that carries the bacterial DNA to other bacteria. Say for example, if this is the phase that packed the bacterial DNA instead of its own DNA, and when this bacteriophage will infect the second bacteria, this bacteriophage we will call it as a transducing phase. Transductant. Transductant means the recombinants formed due to gene transfer by the bacteriophage. That means when this bacteriophage will attack the second bacteria, then second bacteria is known as transductant. So as I already told, this is a rare event. Normally the phage will pack its own DNA only, but sometimes it can pack the bacterial DNA as well. Here the DNA is packed in the phage head. Now phage head is a very small space. So very less amount of DNA can only get packed inside the phage head. So if two genes are present together in the phage head or in the phage chromosome, that means these two genes are located very close to each other, because it is a very small space where DNA can be packed. So only a small amount of DNA is only get packed inside the bacterial phage head. So only the chromosomes that are very close to each other can get transferred and packed inside the viral phage head. Now this transduction, actually it's a two method by which the transduction happens. One is generalized and another one is specialized. Generalized is normal one. The bacteriophage will transfer its DNA inside the bacterial cell. It will replicate, degrade the bacterial DNA, make the phage head particles. So here you can see, instead of packing its own this red DNA, it packed the bacterial DNA, the blue one, and it will transfer this blue DNA into the second bacterium cell. This blue DNA will get entered and recombination will take place between both the DNA, and it will get inserted into the bacterial cell. So this is a generalized transduction. What happens in specialized transduction is this red DNA of the bacteriophage, sometimes it get recombined with the bacterial chromosome. This blue one is this blue DNA. The recombination happens at a site which is called as at site. At means attachment. This site is present both in the phage DNA also and the bacterial DNA also. So because of homologous segment part, it can get recombined in this position. And mostly in the close vicinity of the at site, two genes are present, galactose or biotin. These are the genes that present near to the at site. So now recombination and excision will happen. When the excision will happen, it can again get excised like this, a normal phage DNA, or sometimes after excision is an abnormal phage DNA. That means it now packed the gal site, which is present in the bacterial DNA, this blue section. Correct. So this gal site, it carried the gal plus gene from the bacterial chromosome. But it did not carry the complete gene segment. A gene can only be active or can function properly if from start to end the complete gene segment is present. If the complete gene segment is not present, the gene cannot function. So this is a defective phage DNA now because it packed the gal plus gene but not completely. So after recombination and excision, two situations can happen. A normal phage DNA or an abnormal phage DNA. Now, when an abnormal phage DNA will infect another bacteria, now this abnormal DNA already contained the gal plus. When it will infect another bacteria, the second bacteria also contain gal plus. From the phage, again this gal plus is coming. So what can happen? That this can get inserted, creating a defective copy. Or if that segment does not get inserted into the normal second bacterial chromosome, it will remain as a normal bacterial chromosome. So this normal bacterial chromosome will known as stable transductant, whereas the bacteria containing the copy of prophase will call it as an unstable transductant. So two situations can happen. This is known as specialized transduction because recombination is taking place at a particular position due to which a particular gene, that is gal plus, is getting transferred from one bacterial cell to another bacterial cell. So hence this is called specialized transduction. Now let's solve one question to understand how we can use the process to map the gene. No need to read the complete question because we need only this data to map the gene. The first thing which should be in our mind, that phage head is very small. So the genes that are present in a close association can only get transferred to the phage head together. Now, here the selected marker is leucine plus. With leucine plus, it has been found there are 3 cells that got threonine plus as well, but there are 76 cells that got arabinose gene also. So when looking with respect to leucine gene, it has been found that 3 cells contain threonine gene and 76 cells contain arabinose gene. So from this, it is quite sure that definitely leucine and arabinose are very closely placed genes. When we looking the chromosome on the basis of threonine, with respect to threonine when we are comparing, what we found is 3 leucine genes and 0 arabinose genes. Correct? Here also with leucine and threonine it is 3. So that means threonine and arabinose are not placed in close vicinity. So with threonine, leucine is present, but with threonine, arabinose is not present. This is leucine, this is arabinose, so threonine must have present here. It cannot present here because if it would have been in this position, then the co-transduction with arabinose is possible. But here with the result we saw that threonine has not co-transduced with arabinose. So that means this is not the position where threonine is present. Threonine is present to the place where it is connected with leucine but not arabinose. But it is far from leucine. That's why when leucine is getting transferred, only 3 cells are getting threonine. And when threonine is getting transferred, only 3 cells are getting leucine. But with leucine and arabinose, 76 cells are getting. So that means leucine and arabinose are present in very close vicinity, and threonine is present at some far place but from leucine. So what is the gene order? Threonine, then leucine, and then arabinose. So that's how we can use this data to know the order of the gene. And this question is for you. You have to tell me the frequency of recombination between A and B genes. So if you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.